What is epigenetic rejuvenation?
First of all, epigenetics is the mechanism of gene control that is responsible for turning on genes and turning them off, and also controlling the level of their activity over time or in different tissues. Because, in every different type of tissue, very different genes have to be turned on and turned off. And every cell of our body actually has the full copy of all of our DNA, but the actual genes that have to be activated in a given tissue are very different between, say, your skin cells and your neurons. So what we observe is that the pattern of gene expression changes in a very similar way between people over time. That's why we can talk about this epigenetic clock of aging, or methylation clock, as methylation is one of the epigenetic control mechanisms. So the hypothesis is, if we can bring back the pattern of gene expression to a more youthful state, then we can also expect a more youthful functionality of those systems that the genes are responsible for. Because the genes are what determines the functional state of all the systems in the organisms. This hypothesis is based on the observation that when cells are reprogrammed by Yamanaka factors, they actually exhibit rejuvenation. That if we take a cell from a 100-year-old person and reprogram it using Yamanaka factors to a pluripotent state, basically to an embryonic state, then those cells will also be functionally rejuvenated. And this hypothesis was tested by the group of Juan Carlos Espiso Belmonte from the Salk Institute from California. They did so in 2016. They used Yamanaka factors that they integrated into a transgenic line of mice. There was a certain kind of progeric mice or fast-aging mice. And on those mice, they used the approach of activating those factors intermittently, basically to avoid full reprogramming of those mice, to give short bursts of reprogramming with the hopes that Yamanaka factors, if activated for just 2 days, will be able to rejuvenate the mice, but will not cause their cells to lose their profile of differentiation. Basically, we don't want our skin cells to lose differentiation and turn into pluripotent cells, because the skin cells or the neurons have certain biological functionality that embryonic cells do not. So Belmonte group validated that this approach can prolong lifespan of mice, of those progeric mice, by up to 50%. And moreover, they tested some tissues and some functions of those mice, and they showed that they're actually rejuvenated. That mice treated with that therapy exhibit more youthful state of those tissues or those systems than control group. So this makes us hopeful that this approach of using partial reprogramming can actually turn back our epigenetic clocks, or at least rewind them periodically, so as to help us live much, much longer than currently our genes have condemned us to. So we're working very hard on trying to make this a reality and actually translate this approach into a therapy.